Hi, everybody. My name is Maria Pineda, and I'm the co-founder and CEO of Envisagenics. And today I'm super excited to be here because we're actually using unboxing to present and launch our commercial product, SpliceScore. So I want to tell you a little bit about SpliceScore and what it does and where we started. We are focusing on RNA splicing. We're super interested in this because it's a normal process that happens every day in all of our cells. When this process is mutated or there's an RNA splicing error, it actually causes devastating diseases. Fortunately, we're part of a lab at Cold Spring Harbor in New York that was able to develop a synthetic RNA therapeutic that is actually able to fix and treat spinal muscular atrophy, which is a number one genetic cause of children's death, and it's a devastating disease. So what we realized is that these kids that couldn't move a single muscle were now able to walk with this small RNA therapeutic. And as you can see here, this is a happy story. Now the kids are walking, as you can see in the picture. And my co-founder, while at the lab, actually had developed and used all his machine learning expertise from Technion to develop other drugs like this one for different diseases. So what we did was we licensed out the technology and we started Envisagenics. So today I told you a little bit about that RNA splicing is actually able to solve and fix RNA splicing diseases. So you're probably wondering what RNA splicing is. RNA splicing is how your genomic information is actually translated into proteins. Proteins is what makes our bodies work properly. So for instance, in this sentence, we have a code or a sentence. If your RNA sequence looks similar to this, it actually, in the white letters, could produce a sentence that says, draw a big house on the corner of the white street. And it has a bunch of pink gibberish letters in between. RNA splicing is the process that takes that gibberish out and actually makes a coherent sentence. However, as I said, if there's RNA splicing errors that happens in our bodies, it actually can cause devastating diseases. So in our analogy, we take word number 7 out of the sentence and produces no longer a sentence that makes sense. In our case, if your RNA sequence has an RNA splicing error, it does no longer make a protein. And in doing so, it causes genetic disorders just like SMA and many others. So we developed a software technology at Cold Spring Harbor Lab that allows us to use your genetic code and find these RNA splicing errors. And in doing so, we can then develop RNA therapeutics for them. So I want to introduce you to SpliceScore. It is our artificial intelligence cloud-based platform that allows scientists around the world to do drug discovery. Most of the time, it takes a decade or so, $2.1 billion to develop a drug. SpliceScore, what's trying to do is actually accelerate this process and do it within hours. Most of our competitors' software take 4 to 6 weeks to analyze billions of sequence. It takes us less than 3 hours. So this is one of the examples that allows us to take billions of RNA sequence code and actually narrow it down to a few RNA splicing errors that are causing disease. The example that we have in the screen is around 5 million splicing events. And out of those 5 million, we use our proprietary database that allows us to then narrow it down to a few hundred. Those few hundred are then use AI to actually then understand the biological mechanism and why and how it's causing an RNA splicing disease. And in doing so, by having a handful of targets, then we could try them in the bench, experiment them, and actually design, using our cloud modular platform, an RNA therapeutic, which is a synthetic small molecule that can then treat the patient. And because it is all in silico or in the computer, we're actually able to identify which patients could be used for and actually simulate or model how this drug is going to be used for clinical trials. So RNA therapeutics, it's a growing and exciting market. It's been shown to cause cancer and other genetic disorders, actually affecting at least 30 million people a year in the US alone. It's a $1.2 billion market and growing fastly. For Envisagenics, our go-to-market strategy is 3 different ways. We could license our SpliceCore platform. We could do milestone-based research partnership with biopharmaceutical companies. We could also do in-house R&D discovery to do potential drug targets, all in silico. Because of the success of the drug that was developed at Cold Spring Harbor and the lack of comparable solutions in technology, we've actually have a lot of traction, and we're really happy about that. We now have 7 mutual NDAs with biopharma companies and 4 proof of concepts that allows us to work with some of the largest biopharma companies in the world that are interested in either using our platform to find patient stratification of responders versus not responders, or maybe they're interested in finding new drugs and new treatments for immuno-oncology. At the same time, we're also happy to be partnering with some of the top-tier institutions in New York and some of the early partnerships with one of the largest Japanese pharmaceutical companies. We're doing this because we have a great team at Envisagenics, and we have the support of some of the leading experts in RNA and bioinformatics in the world. We're happy to announce we recently closed our seed round of $2.25 million, and we will actually be raising our A round next year in Q2. And this will allow us to actually complete our strategic partnerships with pharma clients and scale our in-house drug discovery. And with that, I actually want to thank you all for taking the time. If you have questions, find me in the cocktail hour. But of course, if you're interested in AI or finding new drugs for cancer and other genetic disorders, go up to my co-founder up there in the corner. Martine is waiting to demo our platform. Thank you very much.